Okay, so we're going to continue learning about how proteins and other molecules get transported inside the cell. So in the last lecture, we talked about different ways that these molecules can move from one compartment to another. We talked about the many compartments present inside the eukaryotic cell. And we talked about specifically how things go in and out of nucleus, which has the double bilayer, obviously, and then how things move in and out of the membranes of the mitochondria and chloroplast. And then we started talking about vesicular transport. So there are two ways, obviously, that vesicular transport would work. It can allow things to come into the cell, and it can allow things to move out of the cell. When things are moving into the cell, that is called endocytosis. In this case, the molecules or ions or whatever it is that are going to be taken up by the membrane accumulate on the site where they are going to be collected at, and the membrane, the plasma membrane itself, will wrap around them to form a vesicle called an endosome that will then get transported to its location inside. This process is called endocytosis. On the other hand, all day long, our cells are constantly communicating with their environment, with other cells, as well as with different areas of their own, the cell itself, through vesicular transport through a process called exocytosis, where the molecules or proteins will move from sometimes ER all the way up to the outside, or from one of the places in the cytosol. They get carried into these little vesicles that will then merge with the plasma membrane to release their contents outside. So here is an example of the way these molecules can go through. You have your endoplasmic reticulum. The molecules are, when they are formed here, whether they are lipids or they are proteins, they are going to be oriented in that space as they are prepared, so that the part of their surface that is going to interact with the outside of the environment will face the lumen, and the part that is going to be inside the cell is going to face the cytosol. That orientation is maintained all the way through this process as they move from the ER all the way out to the plasma membrane, and then finally to the outside. These molecules are then taken from the endoplasmic reticulum into packages in the form of vesicles that are pinched off with the membrane. These vesicles then go to the Golgi complex or Golgi apparatus, where they can go through further modifications, several steps, or just move through them without any modifications, depending upon what it is that they're meant to do, what their final form is supposed to be, until they're reached to the other side, where transport vesicles would take them out of the cell through exocytosis. Sometimes we see that the proteins that were prepared were not folded properly. Maybe there were some other errors involved so that it didn't work quite well. The molecules were not done correctly. In that case, those endosomes, or those vesicles that are formed, that are pinched off the Golgi apparatus, will be what you would call an endosome. These are different in formulation than what you would see as transport vesicles. These early endosomes would then further sort the molecules inside until they merge with the lysosome, where the remaining contents are then discarded or gotten rid of. So the vesicle budding, one of the ways that this process is controlled is through a special assembly of a protein coat around the vesicle to maintain its structure, its orientation, and to keep their contents the way they're supposed to be. There's a specific set of proteins that work for this process that are adaptin and clathrin. They essentially form a basket-like cage. It looks almost like a soccer ball around the vesicle that is formed to maintain its orientation, to maintain its structure as it moves through the cell to its final destination. So as your product or protein, whatever it is that you're going to be moving, the cargo molecules get assembled at that plasma membrane. They are bound by a protein called adaptin to hold them in their place, to accumulate them in that space. That adaptin molecule is then bound by the Clathrin molecule to create this structure, to kind of form this bud that is then going to eventually turn into the vesicle at the end of the day. It is cleaved by another protein called dynamin that will then release this particular vesicle, so that now this Clathrin coated vesicle will be moving through the cytosol to its final destination. The Clathrin molecules move away as it goes through the system and gets to the final place where it is then going to merge back into the plasma membrane after it is uncoated, so that it can release its content back to the membrane or to the outside. Now, obviously, there are going to be further proteins that help with this process as the vesicle is getting ready to come in and bind back to the plasma membrane, wherever that membrane may be. It may be the plasma membrane, or it may be of the cell, or it may be the mitochondrial membrane, or some other membrane within the body. It could be a lysosome, it could be peroxisome, any of the endomembrane system, so not necessarily mitochondria. So what controls this process are further proteins that are called tethering proteins. These tethering proteins essentially work like long threads that are going to bind to these vesicles to kind of pull them in. In addition, there are other proteins that help in this tethering process. There are proteins that are on the vesicle, that bind to the vesicle first, and those are the ones that are most known for are Rab protein, that the tethering proteins are going to bind to, are recognizing and binding to. And then there are two proteins that communicate with each other. There is a V snare and a T snare. The V snare protein is going to be on the vesicle, while the T snare is going to be on the target membrane. And those two proteins interact with each other to form a dimer of sorts, allowing to pull in that vesicle so that it can bind to the membrane and release the cargo inside the cell. So this is the example of that process being shown here. So the snares are the proteins, specific proteins that will direct this vesicle fusion at the end of the day. The Rab and the tethering protein just hold onto the vesicle to get it pulled into close to the membrane, close enough so that the snare proteins can bind to each other. And then the vesicle versus the target membrane snares will dimerize, bind to each other, get moved away from that vesicle so that the lipid bilayers may fuse. Now, the way this vesicle formation works, this is the typical system that we all know about, right? We know about the endocytosis, exocytosis, that's the one we typically hear about through the endomembrane system. However, there are other ways that secretory pathways work as well outside of these two systems. So before we can talk about that, we're going to talk about how exactly these secretory pathways are controlled and formulated. So in the ER, when the proteins are first made, part of their modifications that are happening at that time are to prepare their tertiary structure, to get them properly folded and formatted, and to properly modify those post-translational modifications are put onto those proteins. These include disulfide bonds that are formed between the cysteine residues to stabilize that tertiary structure of the protein. These disulfide bonds cannot form in the cytosol. The environment is not going to be reducing there. The environment is reducing in the cytosol, so it won't allow these disulfide bonds to be formed. However, in the ER, the environment is different, so that now these structures can form and it can stabilize the protein's tertiary structure. In addition, if the protein is going to require post-translational modifications like additions of sugar molecules, right? If it's a glycoprotein, so then these oligosaccharide side chains are going to be added to these proteins as well. Now, these oligosaccharides can be an important part of their function, but they are also there to protect protein from degradation as they move through these various environments to their final destination. They can help to prepare and stabilize that final tertiary or quaternary structure for that protein molecule to get it into just the right folded stage. And some of these specific sugar molecules can actually act as signaling signaling systems, just like we had those tags in the form of amino acids. You can also have signals in the form of these sugar modifications that allow for appropriate packaging of these proteins into the correct vesicles for final destination as well. Okay, so proteins can, where these proteins are glycosylated is going to depend on its ultimate function and how it's going to be in place at the end of the day. Many proteins that are glycosylated are actually part of the plasma membranes, where these glycosylations stick out towards the outside, because that's one of the ways that cells interact with their environment. They receive signals from other cells, they attach to the extracellular matrix, they are recognized by other cells around them as well. So if you can imagine that these type of sugars molecules are going to be put on the lumen side of the ER, right, on these particular polypeptide chain. They are usually put in place as the protein is getting translated and pushed into the ER itself. So you can see here the amino and the carboxy terminal, the signal that is keeping it, that is moving this into the ER, right? The start signal for that. And then you see that certain lipid linked or just a certain lipid linked oligosaccharides are coming in to bind to the asparagines in the ER, in this polypeptide chain, to attach that sugar molecule to them. So the sugar molecules are going to get added to the asparagine residues inside the polypeptide chain. And you notice that once that sugar is transferred, you see the structure of that polypeptide chain changing to control that protein folding in the appropriate manner. Now, these oligosaccharides are typically found linked to lipid molecules, to fatty acid chains, and then when it is transferred, those fatty acid chains or those lipid molecules will then be left without an addition that is then replenished at a later time. There are some common glycosylations that you can get on these various protein chains. You can have glucose and mannose, which are simple, a little bit more simple sugars, or you can have acetylglucosamine. These are the three most common modifications that are done on the protein chains. Now, again, you still have to make sure that the proteins are getting folded properly, in addition to getting modified appropriately. So chaperones are going to come in and make sure that all the proteins are folded appropriately. Anytime there is a misfolded protein, it is going to be recognized by the chaperone, which will then try to fix it and not move it into a budding vesicle until it has a properly folded protein. When you do have a mutation that causes a protein to be misfolded, or you have some other problem with the protein so that it is misfolded, there are many sensors in place to make sure that that is caught in time before that protein ends up at the location where it's supposed to do work, since it won't be able to perform its work appropriately in its misfolded state. So these sensors are usually in the form of receptors, or that work either singly as a single unit or as dimers, that would recognize whether a protein is folded appropriately or not. different categories of proteins will have different receptors or sensors that are looking for that activated, properly folded protein. Now, if they notice that a protein is inhibited, that a protein is misfolded, they will usually perform some kind of a response to correct that. One of this will include activation of transcription regulators to inhibit protein synthesis until the problem can be solved. Others will activate specific chaperone genes so that you can have the correctly folded protein with the aid of these chaperones. And still other will activate gene expression of other genes that can increase the protein folding capacity so that you can have extra chaperones available, other proteins available, helper proteins available to allow appropriate folding of proteins to form. So once you have the properly folded protein, it is then, and it's modified, it is then sorted based on the signal sequence that is present on there, either in the form of sugars or in the form of actual amino acid sequence, and it is then transported through these vesicles that it's packaged in. When you package these transport vesicles are going to come in, and they are going to fuse with the Golgi apparatus. They will then move through the Golgi depending upon what further modifications are needed, either in a stepwise manner, or they may just move through one membrane to the next until they reach the trans site of the Golgi network, where it will then make its final transport vesicle to go fuse into the outer membrane or another place where they need to go. So here is that example. They fuse at the cis Golgi apparatus. They go through, move through, and get further modified and sorted. And then you have the secretory vesicle getting budded off, where the sorted secreted proteins are going to then go and fuse into the regulated membrane so that that secretion can be done towards the extracellular space. This process is sometimes controlled by the activation of certain receptor through an external signal to begin with. So some signal such as a hormone or a neurotransmitter will bind to the receptor on the plasma membrane. It would induce a signal transduction cascade which would signal the trans-Golgi network to release this secretory vesicle so that it is then fused into the membrane to release outside into the extracellular space. Other times you have proteins that are soluble proteins, but they are not meant for the cytosol, but rather for the secretion into the outside extracellular space, where they will then perform their own function, either right at that cell or some other cell far away. In this case, again, those proteins are going to be prepared and then put into the ER, where they are then those proteins are going to then get incorporated into vesicles and then moved out. These cytosolic proteins many times come in combination, or not the cytosolic proteins, sorry, the soluble proteins many times will come in combination in vesicles that also contain lipid bilayer components. So they may have newly synthesized plasma membrane proteins in there, membrane lipids in that vesicle, so that when they fuse to the membrane, what you end up with is actually replenishing of that membrane space as well. So you have the membrane fusion with these new plasma membrane lipids and proteins, along with secretion of certain proteins that are meant to be secreted to the extracellular space. Now, we're going to talk about a specific type of transport in our eukaryotic cells, which is called endocytosis. Endocytosis is the process where it is internalization of materials that can be taken in by the cells through either phagocytosis or through receptor-mediated endocytosis. It can be a general process or specific to a particular type of molecule or agent that the cell is interacting with. In this way, the phagocytosis can lead to one cell ingesting another cell, or a cell ingesting just small molecules or agents from the outside, depending on what it is that it's doing. One of the best known a process of this is phagocytosis of bacteria or other types of infectious particles in our body by the white blood cells. So the phagocytic white blood cells will engulf these bacteria or other infectious particles with pseudopods coming from their membrane, engulfing them around, and then incorporating them into their own body, where they then destroy them through lysosomes. So in the endocytic pathway, there are fluid as well. Whatever the environment of that cell is, it's going to pick some of that fluid up as well, along with macromolecules or those bacteria that it is taking in. And it's taken in by a process called pinocytosis, because it's a very specific engulfing and taking in of that particular agent into its plasma membrane. It is also used sometimes to balance plasma membrane growth, to kind of replenish it from time to time, to provide it with the new protein receptors or the new environment, based on the new environment that the cells are in. The second one is called receptor-mediated endocytosis, where a receptor that is specific for a particular macromolecule or agent can bind to that molecule and then bring it in through an endosome-like process. An example for this is right here, where you have LDL cholesterol molecules binding to LDL receptors. And again, this is going to take help from Clathrin-coated vesicles. So the Clathrin will, again, just like before, bind around the receptor that is bound to the LDL molecule and create an endosome, a vesicle, which can then get internalized. This is going to then get uncoated and fused with an endosome that's already present inside the cell at that time, and then finally lead to delivery of the LDL cholesterol to lysosome for degradation. As it breaks it down, it's going to produce, using its hydrolytic enzymes, it's going to destroy the basic structure and just release the cholesterol molecules freely into the cell as a result. The endosome can then also continue to, part of that obviously delivered the LDL to lysosomes, but the remaining endosome, the membranes themselves, are going to get recycled back into the plasma membrane, so that those receptors are then ready to work again. So in that process, this is the receptor-mediated endocytosis, where the receptor becomes internalized as part of the vesicle, but then it is then taken back and recycled back out into the extracellular space in that membrane again for further work. So receptor-mediated endocytosis is another example of this. This is another way that our body can fight off infections. So here with virus particles, not only can you, in the other endocytosis we saw that it was engulfing and consuming the bacterial particles. In the case of viruses, it actually can do the opposite, right? So the virus particle use it to their advantage to get into the cell. They bind to the membrane, where these they get engulfed by the plasma membrane and are brought into the cell using these Clathrin-coated vesicles. Once inside, however, they are going to take over the cell to make more viral particles. So, COVID-19 included, this is how many viruses infect cells so that they can continue to grow and do what they need to do. And here you can see that with COVID-19, that you have the spike protein that binds to the ACE2 mediated receptor for ACE2. When it binds to it, that induces that endocytosis through Clathrin-coated endosome. The virus at that point, however, releases its RNA and takes over our ribosomes, uses our ribosome to produce more of themselves so that now it can release them back and cause an infection. So viruses have found a way to kind of take over our body using this system as well. The receptor proteins, obviously, as we talked about, have to be recycled back in this process. So they are going to again be taken in from the cell, right, plasma membrane through the endocytosis, and then from endosomes go back out to be recycled. And that's your typical method that it will move through. Now, some receptors, however, do not get recycled. Instead, they become part of that degradation process as they get taken into the lysosome along with the whatever they were binding to. And in other cases, a transport vesicle may actually take them to the opposite side, the basolateral side of the plasma membrane. So the apical side would be the side that is attached, that is communicating to the outer extracellular membrane. This may be the case in the intestine or in our stomach lining, many places where you are shuttling molecules from nutrients, for example, from one place to another. So you're bringing them into the cell, and then you don't want to bring them back out. You want to take them further into the body. In those cases, what you would see is that the endosome, once that early endosome forms, a vesicle is butted off that will then bind to the basolateral side of the plasma membrane to release its content to the other side of the cell, rather than going back into the plasma membrane on the apical side, which would be interacting with the outside. Now, inside the lysosome, however, when the contents are taken in there, we will find that it is kind of like a mini stomach, right? It has a lot of hydrolytic enzymes that are going to destroy, that can destroy nucleotides, or they have nucleases that would destroy DNA or RNA. They have proteases that would destroy proteins. They have glycosidases that would break down sugars. They have lipases that would break down lipids. So, and then they have phosphatases, sulfatases, phospholipases to destroy phosphate groups, sulfur groups. So a lot of different hydrolytic enzymes, their main job is to break down whatever comes inside. The reason, you will also see that the pH is a lot lower inside these lysosomes compared to the outside. So the pH of a typical lysosome is 5.0, which is a lot lower than what you see in the cytosol at 7.2. It is usually in the plasma membrane or in the membrane, lysosomal membrane. You will find that there are a lot of proton pumps that are bringing in these protons to fuel these reactions using ATP to power this reaction. And then there will be a lot of metabolite transporters that are going to take out those products that these will produce after the breakdown of different things that come into it. So some of this stuff can be recycled. Nucleotides can be recycled, amino acids can be recycled. So anything that can be recycled will be taken back out through the metabolite transporters. Materials that are destined for degradation in lysosome follow many different ways to it, right? And we've already talked about some of them. So one common way is obviously endocytosis. Then you would have that specific type of phagocytosis where an entire maybe bacteria or another thing is going to get engulfed. Another type of cell, maybe a cancer cell even, will get engulfed by the plasma membrane into a phagosome and then taken into the lysosomes. It could also be an autophagy, where the cell itself engulfs its own organelles and targets them for lysosome for destruction. It could be because those organelles are spent or damaged or old, but it could also be in a way to get out of starvation. When cells don't have enough energy or enough nutrients available, they will start to use their own organelles in order to survive. And so that's also a way that this process can happen. So here you'll just see a quick review of these different pathways. Obviously you have, membrane transporters to shuttle things in and out. You have your endosomal recycling, where you have endosomes recycling various molecules into and out of the cell, the receptors included. Then you have the classical secretion pathway, which goes from your nucleus to, the rough ER to these vesicles that merge with the Golgi complex on the cis side, modify these, and then at the trans side form another transport vesicle that merges with the plasma membrane and releases the contents outside. And then you have another kind of non-classical method called exocytosis, where again the molecules are going to collect. This time, instead of collecting on the outside of the cell, they're collecting towards the inside of the cell, on the inner membrane of the cell. And again, there are going to be other proteins that are involved in that process. T-sap6 is one of them. It will form this exosome that will cause membrane flapping and release of the entire exosome outside into the extracellular space. And that is one of those non-classical processes as well. Okay, so with that, we are done with this chapter. And the secretory pathway introduction. We are going to talk more about it later on as we start learning more about how our proteins function inside the cell, how they're regulated through DNA and through modifications over the next few weeks. Okay.